Hello?
Yes.
Can you hear?
Yes, can you hear us? Can you hear us? You announce for the questions. How?
Needed an adapter too, and they gave you that.
Yes, can you hear us?
Yeah, I can hear you. Can you hear me?
Yes, good.
Okay, okay, that's great. That's great.
Maybe you sit here like this. So, they should raise hand. Then our moderator, Simone, on Zoom, and they will. Okay.
Okay.
You could tell.
Hang on, give us just 1 minute. So, hi, so today, so the lecture is going to be live from Beijing. So if you guys have questions, because we probably can't be all in Zoom, our internet's going to support that. Just raise your hand, and then right.
But more you raise the hand on Zoom for China. Okay. And then China interrupts. Right. And then we ask on the microphone.
Yes, sure. But the microphone is not. Microphone is, you know.
But it's everywhere.
Oh, it's everywhere. Oh, that's right. We can unmute it ourselves, right? We can unmute it ourselves. Right?
Yeah, sure. You just press on the thing.
Yeah, okay. You can do it from there. So students, sorry. So if you you have questions, then you raise your hand, and then Simone is going to raise the hand on Zoom. Up the speaker, and then you guys can just ask your questions, because we have mic over there, right? So just say it loudly so it can be heard. Is that okay, Beijing?
Yeah, sounds good.
All right, great, thank you. Yes, let's get started.
Okay, yeah. My name is Sen Song. In case you don't know me yet, yeah. I'm one of the organizers, and I will be introduced Professor Tianming Yang. Tianming Yang is a laboratory head at the Institute of Neuroscience in Shanghai, and he has worked on mechanism of decision making and cognition for many years, using macaque monkeys as a model mostly. And I think Tianming's training is very unique, because he has done experiments, but also thought deeply about deep theoretical questions. You know, his training, he got his PhD degree from College of Medicine under the famous John Mansell. You might know he's a leading scientist in monkey attention field. Then he went on to do a postdoc in the maybe even more famous lab of Mike Shadlin. So Mike Shadlin, you might know, he's famous with Bill Newsom for writing this theoretical paper about the balance network and brain variability. I teach it as one of the classic papers in my computational neuroscience class every year. So I think Tianming has inherited this trait of being straightforward and thinking deeply. So we'll be glad to have Tianming to introduce to us about what's going on in the field of decision making, and hopefully we'll get his personal opinion on how the field is going.
All right, thank you very much, Song. How do I turn on my slides here? Oh, yeah, I can't find my slides. Is it? Is it on this computer or? Sorry. Oh, here.
The camera where we can see this. We just
can you guys see the slides? All right. All right, great. So I'm really happy to be here to share my love for brain with everybody here. And today I'll be talking about the basic frameworks behind the study of neural mechanism of decision making. So unlike previous studies, I would be talking a lot about experiments and how we apply some theoretical framework to these experiments to help us to understand how the brain make decisions. So we know that it's very important for our brain to make the right kind of decisions, right? In the wild, the environment can be really complicated, and it's important for the brain to tell if there's something dangerous in the environment from a very noisy and complicated environment. If you can't do that, you probably will die, and you will not survive in the evolution. But how do we study how the brain makes decisions, especially in the old days? When I'm saying old days, maybe it's just 10 or 20 years ago. All we can do is to stick an electrode into the brain and measure the spiking activity of single neurons. And with this kind of techniques, we do not know how this neuron is connected to the others. We don't have much information about the neural circuitry. All we know is just the spiking activity of this neuron when the animal is making some sort of decisions. And how can we infer from the spiking activity of the neuron there a problem? Oh, I guess you need to share a different screen, right? I see. No. Yes, let's do extension. Oh, there's something we need to fix. No, not this. That one. Yeah. Yeah. Screen. Yeah. Screen 2. Yeah. Okay. I think this is fine now. Yeah. Wait, I cannot move the slide. Oh,
We still don't see our speakers, so if you guys can fix the camera to shoot the speaker, to shoot Tianming, that would be great. Thank you.
You mean you cannot see me or cannot see my slides?
Slides are fine. I'm actually mostly to Pang Hao to shoot the camera so that it's shooting you rather than the student, so we can see you.
Oh.
Oh.
It's a different window.
Oh, yeah. We switch to, but then it's still the speaker, so we have to actually.
Can we?
Oh.
Is it all right? Shall I continue?
Just hang on a moment so we can actually fix it.
Okay.
I see.
Well, I don't think it's very interesting looking at me anyway.
Huh?
Yeah.
Here.
Yeah. Okay, so I guess maybe let's.
Okay, we just Okay, it's good there. Thank you. Continue, please.
Okay, all right. Yeah, so so I guess wait, where was I? Yeah, so so the problem we are facing is that when we can only record from single neurons, how can we infer the neural mechanism behind decision making? All we have is just spiking activity of this neuron, and this is the framework that I'm going to talk about most. So first, I'm going to talk about the signal detection theory, and from the previous lectures, I figure maybe many of you are already quite familiar with the signal detection theory, but I will still introduce it as if you've never heard of it anyway, just in case some of you have never heard of it. So the basic idea is this. If I want to answer the question, is there a signal out there, or it's just purely noise? Let's see. Yeah. How do we do that? And the signal detection theory basically says all you need to do is to calculate some sort of decision variable and compare it against a criterion. If the decision variable is larger than criterion, then we say, yes, there's a signal. If it's not, then we say, no, it's just purely noise. So this sounds like very simple, right? How can this theory help us to detect the signal? How well can we detect the signal? And then now it's and then now let's look at how we can do the analysis. Let's imagine if the noise is Gaussian, if the noise has a Gaussian distribution, and the signal, where's my mouse pointer? And the signal has a different mean, but it's also normally distributed, right? And all we need to tell, and all we need to do is tell whether a particular sample comes from the noise distribution or the signal distribution. And let's just say if the criterion is here, then whenever we see a sample that is larger than the criterion, we will say, yes, there's a signal. So if that sample indeed comes from the signal distribution, and based on this plot, we know the probability of we are correct, meaning we say there's a signal when there's indeed a signal is just the area of this Gaussian distribution on the right side of the criterion. And the probability of we making a mistake, which is called miss in this case, is just the area on the left of the criterion. And the same thing can be done for the noise situation. So if the sample is actually coming from the noise distribution, but it happens to be on the right side of the criterion, we call this a false alarm. And the probability of the false alarm is the area on the right side of the criterion. On the other hand, the probability of the correct rejection, which means we say noise when it's indeed noise, is the area on the left of the criterion. And so based on this, we have four situations, right? We have four outcomes of our decisions. It could be hit or miss if there's indeed a signal. It could be false alarm or correct rejection if there's no signal. And ideally, we want to maximize hit and we want to maximize hit and character rejection, and at the same time, we want to minimize false alarm and miss. But the problem is, no matter how we move the criterion around, the probability of false alarm and hit are always bound to each other. If we move the criterion a little to the right, you can see the probability of the hit goes down, but also the probability of the false alarm also goes down. So it doesn't look like we can set a criterion to an optimal position where we can both maximize hit and minimize false alarm. And if you're wondering if there's any particular position where I can put the criterion so that I can make a universally optimal decision, the answer is actually no. And the reason is that for different kind of decision, actually the cost and benefits associated with each decision outcome can be very different, right? So for example, if the decision is about whether someone's going to have, whether an enemy country will have some sort of mass destruction weapon, then the probability of the false alarm, then the cost of the false alarm may be low, but the cost of the miss could be very high. And in this case, you might want to be on the conservative side. If you have some evidence that the country is really dangerous, maybe you do something bad to them. Well, I'm not advocating you actually do something bad to them, but maybe that's the logic behind certain countries' decision. But on the other hand, if your decision problem is that, well, let's say, if you want to diagnose some kind of disease and the cost of the treatment is very high, then you don't want a high false alarm rate, right? So how you set the criterion actually depends on the benefits and the costs associated with each decision outcome. But there is one single, but there is actually a measurement we can provide independent of how you set the criterion that measures how well the system could tell the signal and noise apart. And that's what we, and that analysis is called the RC analysis. And the idea of RC analysis is actually very simple. We know that the hit rate and false alarm rate are correlated, and it can vary based on how you set the criterion. So if I change the criterion from very low to very high, I should get a serious numbers of hit rate and false alarm rate associated with each each criterion. And the higher the hit rate is, obviously the better your system is. So the deviation of this RC curve away from the diagonal line can be used as a measure of the performance of the system, independent of how you set the criterion. And we often just simply calculate the area under the RC curve, use that as a quantitative, use that as a measurement of the system's performance. Okay. So that's basically how we do analysis on how well a system can tell apart one signal from another. Then how can we use that framework in actual neuroscience studies? So in a series of classical studies, people used an experiment called random dots motion direction discrimination task to study the neurons in monkey's brain, how they can carry information related to this perception, to decision-making task. And in this task, all the monkey needs to do is to tell whether the random dots are moving to a left or right. So the monkey are looking at a bunch of dots. Some of the dots are moving randomly, indicated by the hollow dots here. But some dots are moving coherently, indicated by the black dots here. Of course, in the actual display, you don't see the white and black dots. They are all the same color, but the monkey need to figure out whether the coherently moving dots are toward the left or toward the right. Obviously, if all dots are coherently moving in one direction, the task will be very easy. The signal will be strong. But if only a small proportion of dots are moving coherently, the task will be hard, right? So to give you some idea what the stimuli would look like to monkeys, I will play some movies and you guys can try to play the role of monkey to see if you can get this right or not. Okay. The video will be very short, so pay attention. Let's see. Can you tell the motion of the dots? Yeah, that one should be very easy because the coherence 100%. All dots are moving toward my right and your left. If I lower the coherence, that would be a little harder. On which side? Right, okay, very good. I think all of you have great vision. Well, that one is still relatively easy. About half of the dots are moving toward my left side, your right side. If we further increase the difficulty in this case, let's see how many of you can do this correctly. That? Okay, I can see some different opinion here. In this case, the coherence is 12.8. And the last movie I'm going to show you is really challenging. And I want to see how many of you can get it right. Okay, coming. Right, all right. Nobody is saying left. Are you sure? Okay, let me do this again. Okay, well, actually, that was a tricky question. So in this case, the coherence is actually 0, which means all dots are moving randomly. Nobody, well, it's anybody's guess. And the monkeys are just like you. When there's a good signal, they make good. They make very good choice. When there's no signal, the monkeys are just guessing. So here they, plot the monkey's choice versus the correlation, which is a coherence that I just talked about. And you can see when the correlation or the coherence is high, which in this case only goes up to, I guess, 25. The monkey's performance is already near 100% correct. And when the coherence was at 12.8, and that's when some of you are making mistakes, the monkey is still over 90% correct. And of course, when the correlation is very low, near 0, the monkey is basically just guessing, right? And that's not surprising at all. But now we need to find out if somewhere in the brain there are neurons that can reflect the monkey's performance, because the idea is that the monkeys maybe relying on those neurons to do the task. And where in the brain can we find such neurons? One obvious choice is an area called MT, medial temporal cortex. And For those of you who are not very familiar with the primate visual system, MT is the area where people find lots of neurons that are sensitive to motions. So by saying sensitive to motion, I mean the neuron have different spiking activities when they are when the monkeys are looking at motion in different directions. In this case, we are testing whether the dots are moving to a left and right. If a neuron have different firing rate, spiking activities when the stimulus is moving left or right, then that neuron must be carrying some information regarding the motion direction. And the monkeys may be using that neuron spiking activity for its decision. And that's what we want to find out. So, so these people did the recording, and here is one of the, here is the result from one of the neurons they recorded. And this is the complicated figure. Let's just start from the top row. So, so what they are plotting here is a histogram of the neuron's response. And the black bars are when the neuron is looking at emotion moving towards the direction it doesn't like. We call it the no direction. And the white bar are when the neuron is looking at well, when the monkey is looking at the motion direction that the neuron likes. So when the coherence is high, which is the top row, we can see the distribution for the distributions associated with two motion directions that separated well apart, right? So, so if you remember what I said about the signal detection theory, you can see because these two distributions are well separated, that neuron must be able to tell the difference between left or right very well in this situation, right? And if we look at the other rows, which are the responses to different coherences, you can see as the coherences move, as the coherences goes down, the two distributions gradually moving toward each other, so that eventually they pretty much overlap with each other when the coherence is only 0.8%. So that means in this condition, you will not be able to tell just based on the neuron's activity whether a particular stimulus is moving toward left or toward right, because the two distributions are basically the same. And we can use the SDT to do the RC analysis that I just introduced and plot a curve like this. Okay, yeah. So you can see when the coherence is high, 12.8, the RC curve indicated, it's very hard to see mouse here. Well, anyway, the RC curve is very close to the edge of this box. That means when, even when you have great hit rate, which is the y-axis here, you can have very low false alarm rate, which is the x-axis here. That system would be perfect, would be almost perfect to tell two signals apart. So that neuron can do a great job when the motion coherence is at 12.8. But if you look at the RC curve of 0.8%, you can see basically that curve is on diagonal. That means the hit rate and the false alarm rate is basically the same. So the system, so the neuron's performance is basically a chance, right? So with this RC analysis, we can test how this neuron, we can tell the neuron's performance at different level of stimulus strength. And on the right, that so on the right, we plot the neurons' performance for each stimulus level. And we can see that curve look very much like the behavior curve that I just showed you. So on the right side is the monkey's behavior. On the left side is this neuron's behavior. It's the neuron's performance. The neuron can also do very well when the correlation is high, but at chance level when the correlation is low. And the two, and the performance of the neuron is actually very similar to the performance of the monkey. Indeed, when they look at all the neurons that they recorded, they found the neuron's performance is actually very similar to the monkey's performance at single neuron level. So here the plot is the monkey's behavior threshold on the y-axis and the single neurons threshold on the x-axis. And each dot is just one neuron, right? And you can see most dots lie around the diagonal. And if you plot the histogram of the difference between the two on the top right side, you can see that histogram also centered near 0. So it looks like that not only the neuron's performance is associated with the stimulus strength, which is not strange, but the neuron's performance actually matches the monkey's performance, matches the monkey's performance very well. That, if you think about it, is very strange, right? Here, I'm talking about just one neuron, and we know the monkey have like, well, I don't know exactly how many neurons are in the empty area, but there are a lot. But if single neuron's performance is already as good as the monkey's performance, what are all the other neurons doing? And that question, well, we still don't have a very good answer. There may be a coincidental finding, but how can we relate the single neurons performance to a population, to a population of neurons performance? One simple hypothesis is that the population is just a linear average of single neuron, right? So in this case, if we consider a single neuron, each single neuron conveys some information of the sensory stimulus plus some noise. When you adding them together, do an average, the noise should be canceling out. So the population should have much better performance compared to single neurons. But that doesn't seem to be the case, at least in the study that I show you. And why? One possible reason is that there are correlations. There are two kinds of correlations that we usually talk about when we talk about the neuron responses correlation. One is what we call the signal correlation. So here, for example, if I record from two neurons, neuron 1 and neuron 2, each are tuned to the motion direction, so that when the motion is upward, both neurons have larger firing rate. When the motion is downward, both neurons have low firing rates. Okay, so if you measure the neuron response across a wide range of motion, you will see two neurons' firing rates are correlated. But that correlation is mostly due to the fact that these two neurons have the same, have very similar tuning properties regarding the motion direction. So that correlation could be explained by the signal. That's what we call the signal correlation. But in addition to signal correlation, we also find even at the same, even when we use the same signal, the neurons firing rate still varies from trial to trial. And that variation, and the correlation due to that variation is what we call the noise correlation. So, so here on the left, I'm just trying to indicate the trial by trial variation with this bunch of dots, and these two group of responses are based on the same stimulus. If we plot the neuron 2's responses versus neuron 1's responses, we sometimes get a figure like this. So clearly you can see this is the situation when the two neurons are looking at exactly the same stimulus. The correlation between the two neurons cannot be explained with the signal. And it's due to what we call noise, but it could be something within the system or something external that we don't measure. But in any case, we would group this as we will call this noise correlation. And of course, if you don't see any noise correlation, that would be the case when the two neurons firing are completely independent of each other. Okay, so the natural question is, how does this response correlation would affect the population performance? Actually, my plan is I will not tell you about this. That's part of the homework. Well, it's actually a very straightforward application of the SDT. So I think you should try to figure out how noise correlation could affect population performance. And you could consider variables like population size, of course, noise correlation itself. And in another case, you can consider the case when you are only when the downstream is only reading a part of the population of the neurons for its decision making. What would happen in this case? So anyway, this concludes my introduction of the SDT. Now back to the black box question. Here we have seen that how we can measure the neurons ability to tell apart two signals, but we haven't really say anything about whether these neurons are actually contributing to the choice or not, right? We have only looked at the left part of this diagram, but not the right part. So if we want to ask the question, how do these neurons contribute to the action? What can we do? One possibility is that we can use SDT again to measure the neurons' contribution to behavior, and this is what we call the choice probability analysis. And the idea is very simple. So instead of looking at the neurons' response to two different signals, here I'm measuring the neurons' response to two different behavior. So when the monkey is making choice A, I plot the distribution of the neuron. And when the monkey's choice is B, I plot another distribution of the neuron. And naturally, if I do RC analysis and use that measurement, I can tell whether just based on the neurons fire rate, can you predict if the monkey is going to make choice A or choice B, right? so that's a basic logic behind choice probability, and that logic may be faulty because this is a purely correlational study, right? So you're trying to correlate the neuron's firing rate to behavior, and that correlation does not guarantee causality. So, but it's still a very interesting quantity that people still use very often in their studies. It could at least provide us hints about neurons in certain brain areas and how they may contribute to certain behavior. So here I'm leaving the second thinking point of the lecture, like, basically I would like you to find out how choice probability can be related to the same variable that we have already studied with, and that I have already mentioned before. Okay, so in summary, now we have a framework that can help us to understand how neurons encode information and how they may contribute to decision making, and that framework only requires to use the spiking activity of one single neuron to do this. And although it's very simple, but it's very powerful, and it can tell us a lot of information about how neurons carry information and that kind of stuff. But still there seems to be something missing, because when we are talking about how we make decisions, we are not only consider what kind of decision we are going to make. We are not only just considering how likely I can be right or wrong, right? There's a whole different dimension that's not even mentioned in the SDT framework, which is the time. Time is very important for decision making, right? In some cases, when we have plenty of time, we can try, we can fine tune our decision making, try to be as accurate as possible. But in other situations, we have to make a very hasty decision, which can be wrong. But if we do not have enough time, that may be all we can depend on. So how can we incorporate time as part of the framework to explain our decision making? And people study that, again, with monkeys, again, with random dots. So in this experiment, it's very similar to the random dots experiment that I talk about. The only difference is that they no longer showed monkey a movie with a fixed duration. Instead, the monkey gets to decide how long they would like to view the movie, and the monkey can indicate their choice anytime during the motion viewing period, and we can measure the time between the onset of the motion stimulus and the onset of the monkey's choice behavior, and use that as the reaction time of the monkey. So so reaction time indicates how so reaction time is an indication of how much time the monkey needs for making its decision. So so naturally, reaction time is also dependent on the motion strength, right? if the task is very difficult, the motion strength is very low, the monkey would take a lot of time to make its decisions. But if the coherence is high, the task is very easy, the monkey would only require very little time to make its decision. So, so overall, the percentage of correct still look. So overall, the accuracy of the monkey still, well, in the reaction time task is still pretty similar to the fixed duration task, but it's a reaction time measure that that we want to explain with the new framework, and that framework is called the Drift-Diffusion model. So the idea of Drift-Diffusion model is basically like this. So we will model the decision-making process as a particle moving between two decision bounds. So here in this diagram, the x-axis is time, okay. And at time 0, that particle starts from 0 point, and then it would drift toward one of the two bounds. And whenever it reaches one of the bounds, a decision is made according to the bound that's to the bound that's hit. And how does the particle drift? Well, at each point, the system would receive some evidence, and that evidence is what we call the momentary evidence. And that evidence is distributed. Well, we can assume that evidence is again distributed normally with a certain mean and the standard deviation. And if the mean value of if the mean value of the momentary evidence is far away from 0, let's say it's positive, obviously that that evidence gets accumulated over time, what drives the particle upwards towards the upper bound, right? And the time from 0 to the time when the particle reaches the upper bound is the reaction time. Okay, so that's a basic idea of the DDM model. And
if you want,
okay,
depends on you. Yeah, you can finish this slide and then.
Okay, maybe let me finish yeah, I will take 5 more minutes. Okay, so so obviously how soon you reach the bound depends on the quality of the evidence, right? I mentioned if the evidence is strong, the mean of evidence is far away from 0 line, you can accumulate the evidence much faster and reach the bound much faster. So that would be able to explain the reaction time that we saw in the experiments. And in another case, when the evidence is weak, then because of the noise, the particle could take a very long time to reach the bound. And you can actually you can actually calculate the percentage. Well, you can actually calculate the performance of the model based on the bound height and also the distribution of the momentary evidence with the equation on the right side. If you're interested in how we get this equation, you can read the article. It's basically using the martingale but I don't think it's very important as long as you understand that we can calculate just the both the performance of the model and also the reaction time of the model based on the bond height and the features of and the feature of momentary evidence. Okay, so I think let's take a break now, and we can come back.
Yeah, let's take a 10 minutes break.
10 minutes, okay.
I guess, yeah, 10 minutes break. I think the Basel side, this you can see the students are still discussing. Wait a little bit more. The rabbi is just coming in. Yes. Yeah, let's start. Yeah. Okay, let's start.
All right. Can the guys online hear me?
Can you hear Tianming? Basil, I think your speaker is micro is turning off.
Yes, we can hear you.
Okay, great.
Great.
All right. I will continue. So I just introduced the DDM, and now we can see how well DDM works. And on this slide, it's a fit to the monkey's behavior, and you can see the black dots is the actual data. So they measure the monkey's choice as well as the monkey's reaction time, and the black dots are the actual data, and the black line is the DDM's fit. So you can see the DDM can explain both the choice of the monkey and the reaction time of the monkey really well in this case. So now we have a model that could explain monkey behavior, and the next thing is we want to find out if the brain is actually using a mechanism similar to DDM to solve the task. And when we go to the brain, we are trying to find a brain area that's that can integrate the motion information to make decision making. And we already know that MT area encodes the motion. The MT area carries the motion information for decision making, and anatomically, there's an area called LIP in the posterior parietal cortex, which receives a lot of projections from MT. So people guess maybe that's the area where the information goes and makes the decision based on the sensory information. So So LIP is the target brain area we will be looking at. And there is something interesting about the about the response property of LIP neurons. So LIP, people have found, is involved in planning eye movements. Okay. So I didn't mention that in that particular random dots experiment, the monkeys were asked to move their eyes to indicate their choice. So when the monkey thinks the dots are moving to a left, the monkey should move their eyes towards the left. And when the monkey thinks the dots are moving to a right, they should move their eyes to a right. Okay, so the answer is indicated by monkeys' eye movements. And the LIP has been found that it's involved in planning eye movements. So when I say planning, that means that the neuron would become active even before the monkey actually makes the eye move. And the neuron have this property called response field. Here, here I use this gray region to indicate the response field of a neuron. If the monkey is planning an eye movement toward a target, toward an eye movement target in the response field, the neuron would become active. Okay. So here is a summary of their finding. What they found is that where's my mouse pointer? Okay, yeah. So they plot the LIP neurons responses when the monkey is looking at the motion stimuli. And what they found is that the LIP neurons, so they found that the fire rates of the LIP neurons go up very quickly when the signal is very strong, when the motion coherence is large. That's the orange curve here. And when the motion strength is low, like the blue curve here, the neuron's fire rate goes up much slowly. And all these solid traces are the case when the monkeys would eventually choose a target in the neuron's response field, right? So that means that if the monkey is going to choose the response, if the monkey is going to choose the target that the neurons like, the teeing target, the target in the response field, the LIP neurons responses will go up during the time when the monkeys are looking at the stimulus. But how fast it go up depends on the signal strength. But on the other hand, if the monkey would eventually choose a target outside of the response field, the TL target, the neuron's response would actually go down. And that's a dotted line here. So, by the time the monkey actually indicates its choice with the eye movement, we can see there is a very large separation between the solid traces and the dotted traces. So the solid traces are when the monkey chooses a team target, and here you no longer see the difference between different motion coherences. All the choices merge together to reach the it looks like all the traces merging together to reach a threshold before the eye movement actually happens. But on the other hand, when the neurons, when the monkey is choosing the TL choice, the traces don't exactly merge together. So this is as if this neuron are doing the DDM. They are accumulating information regarding the teeing target. If the signal is very strong, the you would accumulate the information much faster, and this neuron fire rate will go up much faster. But eventually, when you hit the bond, all the traces merge together, right? In contrast, if we look at the MT neuron responses, which is plotted down there, you can see a relatively flat trace during the time when the monkey is looking at the motion stimulus, right? So, and the higher trace is associated with this strong signal, but that trace does not really go up. So the information doesn't seem to be accumulated in the MT area. MT responses only seem to reflect the quality of the stimulus. It is the LIP where neurons responses reflect reflect the evidence accumulation process. Okay, so here we are coming to the thinking point number 3. After the lecture, I would like you guys to try to think about if I have a population of neurons doing DDM, and I have let's say let's say some sort of weighted average mechanism to read out the population of neurons. How what does the choice probability of these neurons will look like in this situation? Again, you can consider the factors such as population size, noise correlation, so on and so forth. Okay, so I just introduced DDM in the random dots experiment, but actually people have found the DDM can be used in many other decision-making tasks as well. So, so for example, value-based decision-making or time perception, memory retrieval, so on and so forth. And I will talk about probabilistic reasoning in this case, because I think this will tell us what exactly is happening in DDM. So when I was introducing the DDM, I said it models the decision as a process in which a particle is driven by evidence, right? But the evidence in this case is actually not well defined. I did not actually say how you can compute or quantify evidence in this case. But there is a, but in statistics, there is this sequential probability ratio test, which is actually very similar to DDM. Here, we also have 2 decision bounds, and we are accumulating evidence during the sequential probability ratio test. And what exactly is being accumulated in sequential probability ratio test is what we call log likelihood ratio. And what's log likelihood ratio? So log likelihood ratio is basically the log ratio between 2 conditional probabilities. When you observe some sensory evidence, you consider these 2 probabilities. What's the probability I observe this piece of evidence if hypothesis 1 is true? And what's the probability of I see the same evidence if the hypothesis 2 is true, right? So intuitively, if I'm more likely to see this piece of evidence when hypothesis 1 is true, then this piece of evidence should support hypothesis 1, right? So the probability of seeing this, so the probability ratio between, so the ratio between these 2 probabilities should be larger than 1, and and the log ratio should be positive, and that piece of evidence would bring the particle a little upward toward the upper bound, right? So, so that seems to be a very good explanation of DDM. So if we can quantify evidence in terms of log likelihood ratio, then we can use that to calculate how well the DD model could make decision based on probabilistic information, and we can further find out if the brain is really using this sequential probability ratio test to do the decision making. But how do we prove this? We cannot really prove this with the random dots experiment. The problem is that the random dots experiment uses the random dots, which is very noisy by itself. And in addition, our visual system would introduce further noise when the system processes the motion information, and those internal noise is very hard to quantify. So it's very hard to calculate this conditional probability. To find out, so we need something different to find out if the brain is doing this probabilistic probabilistic reasoning process, and here's the task we designed. So in this task, we asked the monkey to make a decision between the red dots and the green dots based on a sequence of 4 shapes. And after viewing 4 shapes, the monkey again uses its eye movement to tell us whether it chooses the red or the green dots. And because the shapes are designed by us, we can actually specify exactly how much information is conveyed by this shape. So the way we do this is we would assign weights to each shape, and the weights would indicate whether each shape is a piece of evidence in favor of the red or in favor of the green. And the weight is actually the log likelihood ratio. That and the conditional probability here is like this. So even if a given shape is presented when the actual answer is red, versus we see the same shape when the actual answer is green. So if the probability ratio, well, if the log likelihood ratio is positive, that means that shape is piece of evidence in favor of the red choice, right? And vice versa. So by assigning weights to each shape, we can train the monkey to learn how to make choice based on these shapes. And the monkey can actually do this task. So if the monkey learns the task perfectly, what does the monkey has to do if he sees these 4 shapes? All he has to do is just to add the weights associated with these 4 shapes together to a number which is 0.2 in this case, and then convert this number to the reward probability of the red and green dots. And if the red is larger, it should choose that target as the correct answer. Okay, so that's a task. But of course, the monkey has no idea how this works, right? He has to figure out just by trying, like when I see the triangle, is it more likely to get a reward from the red target or the green target? So he has to learn this trial by trial. So through a lot of learning, he actually finally learned this. So here I'm plotting the monkey's choice, the percentage of red choice versus the log likelihood ratio. So you can see when the log likelihood ratio is positive, a large positive number, the monkey almost always choose the red choice, and vice versa. And when the task is high, when the task difficulty is high, it's very hard to move the mouse. Okay, when the task difficulty is high, meaning the log likelihood ratio very close to 0, the monkey choice varies a lot between red and green. That's natural. That's just when the monkey is dealing with very fuzzy information, right? But at least we know that the monkey is using the combined log likelihood ratio to make this to do this task. And after we train the monkey, we go back to LIP to see whether LIP neurons are indeed calculating those log likelihood ratio. And here I'm going to show you some short movies that that I made to when the monkeys are doing the task. So so what you will be seeing is the task itself. Just as a monkey, you will see 4 shapes appearing on the screen, a red choice dot and a green choice dot. But when we are recording from the neuron, one of the dot is in the response field of the neuron. So if the monkey is eventually to choose the red target, in this case, the neuron should be very active. Okay, let's go. So you can see 1, 2, 3, 4 shapes. And by the end, the monkey chooses the red target. And to help you understand what's going on in this task, I plot the log likelihood ratio at the bottom of the screen. So see if I can get this. Okay, so we have four shapes, right? So the first shape is in favor of the green target. The log likelihood ratio is negative, and the second shape is in favor of the red target. So that brings the total log likelihood ratio towards the positive side. And the third shape again is in favor of the red target. So the total log likelihood ratio goes even further up. And finally, the monkey chooses the red target. And in addition, I'm also plotting the spike train as a short white bar here. And you can see in the beginning that particular neuron has a low firing rate. But as the shapes coming up and the monkey is accumulating more and more evidence towards the red target, which is in the response field, the neuron become more and more active. And eventually when the monkey chooses the red target, that neuron's response would reach a peak. Okay, so I can play the video again so you can see how this works. And we have a different, I have a different video to show the opposite case. In this case, the monkey eventually choose a green target. And because the green target is not in the response field, so the neuron's firing rate becomes lower and lower as the evidence accumulates towards the green target. And we also have cases in which the choice actually switches. So in this case, the evidence is first in favor of the red target, then the second and the third shapes bring the evidence towards the green target, but the last shape is actually a strong red shape target, red shape evidence, and the total log likelihood ratio gets reversed. And you can see the neuron's response first go up, then go down in the end, right? And I have another I have another example showing exactly the opposite case. The evidence first goes against the target in the response field.
How difficult it is to train the monkey to do this?
Well,
to do this kind of task?
Well, yes. So the training takes a long time, obviously, but it's actually not that difficult. So we first train the monkey to use just one shape to do this task, and afterwards we show monkey 2 shapes, 3 shapes, 4 shapes, building this up gradually, and yeah, so that's basically how we train the monkey.
Professor Yang, I'm curious that why do you believe that the monkey or the LIP is doing exactly hypothesis testing instead of something like reinforcement learning, like accumulating just reward or something like that? Which means that they are not doing inference at all.
Yeah, this is a very, very good question. As you noticed, the monkey, well, maybe let me finish introducing the LIP part first. Then I can come back to your question. Yeah. So by looking at these movies, you probably get an idea that this LIP neuron can tell me what the monkey is thinking about during during its decision making, right? So, for example, even for example, even if I just show monkey 3 shapes and the monkey hasn't told me anything yet, just by looking at this neuron, I know that the monkey is probably going to choose a red target if I stop the shapes by that time, right? But that's not enough. We still want to know if the LIP response would exactly tell us the log likelihood ratio. So what we do is we will plot LIP's responses by the log likelihood ratio, and you can see the neurons have higher firing rate when the log likelihood ratio is high, lower firing rate when the log likelihood ratio is low. And if we calculate the average response after shape appears, we can do a plot like this, right? So the neurons' responses seem to correlate with log likelihood ratio very well. And we can do this for each shape. And we found for especially for the 2nd, 3rd, and 4th shape, they all have a very, very similar relationship between the neurons' responses and the log likelihood ratio. The 1st shape is a little different because we have a very large response to the target onset itself. So the signal we see in so the response of LIP we see during the 1st shape presentation is contaminated with other responses. But in the other cases, no matter how many shapes monkeys see, LIP responses seem to be faithfully reflecting the total log likelihood ratio for decision making. So so we seem to find that the brain is indeed calculating log likelihood ratio during evidence accumulation, and the LIP seems to be the exact area where neurons are doing this calculation. But people might still argue that maybe the signal we see LIP is not computed in LIP itself. It could be coming from somewhere else, right? we know that a lot of brain areas may be involved in decision making. There are several other studies even showed very similar ramping activity in other brain regions. Why would I say LIP? Why would I believe LIP is special among all these areas? So we did another study, and in this study, we made the task even harder for monkeys. And the way we do the new task is like this. Again, we train a monkey to make decision between a red and green target, but now the shape itself has color as well. So for example, it could be a red square or a green triangle. And the monkey has to accumulate information for the red target only using the red shapes. And the reward information of the green target is only conveyed by the green shapes. And we show monkey totally 6 shapes, so that the monkey has to do even more computation. So in the old experiment, all the monkey has to do is just to accumulate information associated with each shape, right? But now, not only the monkey has to accumulating information associated with each shape, it also has to consider what color is that shape, right? So, for example, in this case, a square in red could be evidence toward the red target, but the square in green is actually the evidence against the red target. So the monkey need to combine the shape information with the color information, then accumulate the evidence using this combined information. And our idea is that we want to find out where the monkey would combine the shape and the color information together before the actual evidence accumulation happens, right? And if that computation step also occurs in LIP, then probably LIP is where the evidence accumulation happens. So that's the logic behind our reasoning. And so first of all, I have to show you the monkey actually knows that task, right? So here, so in this new task, the monkey's choice should depend on the weights of the red shapes and the rates of the green shape. So here the x-axis is the difference between the red weights and the green weights, and the y-axis is the probability of monkey choosing red target, and you can see the monkey is doing this pretty well. And furthermore, we can use logistic regression to figure out how each shape affects the monkey's performance. So here the positive value just means when this shape appears, the monkey tend to choose the target with the same color. Okay, so if it's a red square, the monkey tends to choose red. If it's a green square, the monkey tends to choose green. And because both red square and green square has a very high weight, the monkey would think this a good piece of evidence. But when a shape has negative weight, it's piece of evidence against the same color. So the logistic regression would give you a negative number. And there's a fact that the logistic regression give you the coefficients that matches our assigned weight, shows that the monkey actually understand how they can make choice based on each individual shape. And this is very similar to the previous analysis, basically just showing you LIP is accumulating information based on the log likelihood ratio. So I'm not going to repeat on this, but But one interesting point I want to point out is that if you look at the response after the onset of each shape, wait, where's my mouse? Okay, so for example, this axis is the onset of the fifth shape. If you look at the response after this onset, there seems to be a lot of dynamics going on. It's not just going up. It actually first goes down, then goes up. And we wonder, what does this dynamics mean? Does it tell us something about how monkey combines the shape and color information together to calculate the evidence that's ready for accumulation? Okay, so we hypothesize that the monkey probably can use one of the two routes to do this task. Imagine the monkey again is seeing a red square as evidence, and the green target is in the response field. The monkey could first use the weight of the square and combine this, combine the color of this shape, red, to calculate the color evidence. So the color evidence just means now we have the evidence for the red is 0.5. Then based on whether the green target or the red target is in the response field, then I can calculate the evidence for the response field target, the T target, right? Another way to calculate the evidence is that first I use the target configuration to figure out whether the current shape should be considered as the evidence for the T-in target or the T-out target. So that's what we call the color consistency. So if the color is consistent, then I can use this weight directly to calculate evidence. If the color is inconsistent, then I should change the sign of the weight before it can be accumulated, right? So regardless how you do the calculation, before you finally get the evidence that's ready for accumulation, you have to carry out multiple steps of a computation. And how can we find out if these steps are okay?
So I wonder, the fine rate after the onset of maybe one shape, the fine rate first go down. Does that mean that one route is faster than the other route?
Yeah. Yeah, you can hold this question. Well, I will explain how we try to figure out.
Thanks.
Yeah. Well, it's not exactly right, but the intuition is correct. Well,
thanks.
Yeah. So, yeah. So I mentioned that that there could be a lot of computation going on in this task, right? And the shape keeps and the shapes keeping and the shapes keep coming up one by one very quickly. So so a lot of computation may be overlapping in time, and the representation of this computation could also be overlapping in time. And how this computation could affect the neuron's response maybe also quite complicated. So we use this Poisson GLM, try to dissect the influence associated with each event. And the basic idea is that for any particular input to the system, it might affect the response over a period of time. like this. So for example, if the onset of a particular event is here, then it first increases the response, then the response quietly dies down. So so the influence of that particular event would last over a period of time. Then during this period of time, if there is a second event coming in, then you can combine the influence of these events together, and mathematically you are just doing the convolution between two things, which is the kernel of each event and the time sequence of the events. So the time sequence of the events is X, and in our case is, well, and the kernel of these events is K here, and we also have to assume that the neuron have a certain mechanism of firing spikes based on the kernels, and as usual, we assume the neurons are Poisson, meaning that there is some latent state of the neuron that determines the firing rates of the neuron, and that latent state is a lambda, which we can compute based on the kernel and the event sequence. Okay, by doing a convolution, and the events in this experiment could be all the variables that we are interested in, like the weight of the shape and whether the color matches the gene target, the color consistency, so on and so forth. And
in the last equation,
delta. Oh, here. Oh, here we are just considering. So this delta is just the time window. Okay, so yeah. So so this delta lambda t is just the latent state within this within the t's time window. Okay. Right. So all we need to find out is how these variables affect the neuron's response. So so we just need to plot the kernel that are related to each variable we're interested in. And the first one we look at is the weight of the shape. And we found most neurons have a negative weight of shape. So on the left, in this heat map, each line is one neuron, and the color indicates the kernel weight. If you see a blue color, cool color means negative weights, and and you can see most of the neuron have a negative weights. If you calculate the average weights of the population, that's the right side of figure. Overall, the neuron seems to overall the kernel associated with the weight have a very small latency. Trying to figure out where is the mouse point. Yeah. So the kernel of the kernel for the weight have a small latency, but it is very negative initially. Afterwards, it go back to 0, and that seems to be consistent with the computation that I just introduced. You only need the weight information initially in the first step of the computation. When you come to the step where you need to accumulate the evidence, that weighted information is no longer necessary. And that seems to be the case for this LIP neuron. But if you look at the evidence kernel, so here the evidence is after you combine all the information, the color, shape together to calculate whether that particular shape is evidence for the T-in target or T-out target. If you look at that kernel, you found it's mostly positive. So positive weights means that's evidence in favor of the T-in target would increase the LIP responses, right? So in this case, that's very consistent with what we know about LIP neuron. If you receive evidence in favor of the T-in target, the neuron's response would increase. And
there's a question on chat room. Yuxing has technical questions, I guess. If neuron data was calcium rather than spike train, which kind of linking function should we choose for GLM?
Calcium signals. Okay. Yeah, well, yeah, I think you can use the different linking functions, of course, because calcium signal, well, people have been using using very similar convolution process to deduce the spiking rate based on calcium signal. So I guess you can do something quite similar. If you get the calcium signal, you can first go back to you can first convert it back to spikes. Then you can use the same kind of GLM model, or you can just well, I don't know if anybody has actually directly used the GLM for calcium signal, but I think there should be a way to do this. Yeah, it's a good question. I think it can be easily solved, but I haven't done it before. Okay, so
I'm a little bit lost here. The kernel weights and the there's the previous one. This is a cumulative evidence kernel, and the previous one is
is the weight kernel. Yeah. So
how are they combined in your model? In the GLM.
Right. So here basically we set the onset of the weight and the onset of the evidence to be the same, right? So so because all this information theoretically should be ready when the shape is presented on the screen, and but we don't know when it would actually affect the neuron's responses. So so when we do the GLM, we just use the same time for all these variables, and the latency of these kernels could tell us how when this variable actually affects the neurons' responses, and that would be the indication of when this computation actually happens.
Basically, for each shape, you have two different kernels. Is that
actually much more?
Much more.
Like shape, weight, evidence, consistency. Basically all the intermediate variables over there.
Okay.
Yeah. So yeah. So the evidence kernel is actually what we all know about LIP. It just increased the LIP's responses according to the evidence, and also you probably noticed that the evidence kernel is sustained. It doesn't go back to the zero line as a weight kernel. And that's also consistent with our idea that evidence has to be maintained and accumulated. And we also looked at the other intermediate variables, but it turns out the result is different in two monkeys. So in one monkey, we found a very strong color consistency kernel. And in another monkey, we found a very, very strong color evidence kernel. And that seems to be suggesting that two monkeys are doing two different computations. So if we come back to this figure, you can see that the color, so you can see the color evidence signal is only important for the route 2, but the consistency signal is only important for route 1. So, but regardless which route you choose, the results seem to indicate that these intermediate variables are represented in LIP. And if you choose one route, the other intermediate variable is not represented in LIP. So I think this means the finding is not by chance. If any possible variables you can find here, it's it would argue against the idea the monkey where the neurons are doing the calculation we think they are doing. And in addition, as I just hinted in the previous slides, we can measure the latency of each kernel. And the latency of each kernel is also consistent with the computation steps. So for one monkey that that computes the consistency, we found the latency of the kernel for weight is the earliest, then consistency is intermediate, but the latency for the final evidence is the longest in this case. And the most interesting thing is that these kernels, if you compute it for each individual neuron, seems to correlate very well. That means if a neuron encodes one variable, let's say weight, it also encodes the other variable well, let's say consistency. So there's a very good correlation between the encoding of these variables. If the neuron is involved in first computational step, it's also involved in the second computational step. It's not different populations of neurons doing different computation steps in this case. Okay, so in the end, our results seems to indicate that the monkeys, well, the neurons at least, their responses reflect the computation steps in this complicated evidence accumulation test. Two monkeys seem to be using different strategies, but the overall computation process seems to be consistent. And that seems to suggest that LIP neuron not only is doing the evidence accumulation, it's also doing computation related to evidence accumulation. And there are other studies seem to suggest that those weight and the color information may come from the prefrontal areas or the visual areas in the ventral stream. And the final and the accumulated evidence of LIP would reflect the monkey's final decision in the end. Okay, so I've talked a lot about the DDM. how we find the LIP response reflect the DDM process. And finally, I talk about our recent finding that the neurons not only reflect the DDM itself, but it also reflects the computation in addition to DDM. So here comes to the final thinking points that I would like to ask you guys to think about. So still, our finding is a correlational study, right? We used Poisson and try to dissect each component of LIP's responses. But if I want to do some causality experiments, although I know most of you guys are here to learn some theory, I will challenge you to think about an experiment that can help us to prove or disprove our conclusion. Is LIP's evidence accumulation actually happening within LIP or not? Okay, so that's all I have to say. I hope I
just in time.
Okay, great.
Just in time. All right, I think
ready for questions.
Yeah. Any questions that there's an inference question previously? Maybe you want to.
Right.
Yeah.
Wait, can you repeat your question again?
Yeah,
sorry.
I guess that question is not easy to answer. So the question is, how can you prove that the LIP is doing inference exactly? Because if we are thinking about reinforcement learning, it can be some signal like Q of the item and action, right? The Q value in the reinforcement learning. Maybe that's what is accumulating.
Right, right. Yes. Yes. That's a great question. That's actually a very good discussion point for all these kind of experiment design, right? We have a model. The model could explain the behavior very well, but that's not the only model that could explain that behavior. There could be many different models, and for the time being, we find some neural correlates of the model that seems to be consistent with the idea the brain is using this model to do computation. But that's not the proof that the brain is not using the other model. That's a weakness. There's a logical weakness here, but in general, when we have a model, how can I argue that this is a good model, a reasonable model? Then usually the argument is as follows. First, the model should satisfy certain kinds of optimality. So meaning that the model should solve the decision-making task well, right? It should make sense in the context. So in case of the DDM model, it has been proved that the sequential probability ratio test is the most powerful test. It takes the least steps it takes the least steps to reach your designed performance, your designed accuracy level. So based on this, you can argue that model makes sense for this kind of task. This is the first point, and the second point is, and the second argument that the model is a good model is that the model matches the behavior well, and we can find the neural correlates of the model well. And and that's also true for DDM. Many good models are like this. They first make sense, and they can both explain the behavior and the neural data very well. But as you mentioned, that's not the definite proof how brain does the computation. This is a very difficult question. This is very difficult to prove unless we have complete knowledge of how neurons are wired together, how information are passed around between the neurons. We won't be able to answer this question for sure, 100%. So for the time being, I think all we are looking for is a model that makes sense and explain behavior and neural data well.
Side, and I think you guys have been very quiet, and we haven't heard anything from the chat room, and we don't know any questions or any comments, please. Yes, I see someone raise their hand. That's great.
That was okay. Yes, but we do have questions. So I think we're going to. Yeah, can you hear us?
Yes, we can.
Okay, go ahead. Question. Go ahead and yeah.
Hi, Professor Yang. Thank you very much for the presentation. I wanted to ask, did you always only look at a single neuron for evidence accumulation, or might there be multiple neurons that do the same job?
Same neuron doing the accumulation, or multiple neurons doing the accumulation? That's your question,
yes.
Right?
Yes.
Right.
There's a special
yes.
mechanism.
That's also a very good question, because we don't have a good answer. It's kind of situation like the experiment I talk about in the signal detection theory experiment, where we show that single MT neuron could match the monkey's performance. Here we find is actually something quite similar. Single LIP neuron could account for monkey's performance largely, and obviously there are a lot of LIP neurons. Many of them are doing similar things. We do not know how the brain would combine the responses of all these neurons to draw a conclusion, to make a decision. All we know for now is that even at the single neuron level, you could explain the brain, the monkey's decision making pretty well.
I think we have another question. Go ahead, James.
I have actually two questions.
Right.
For the random dust task, what's the role of noise correlation in this kind of computation? And what's the role of noise correlation in when you were talking about the random dust task? And then for the last part of the talk, do you think like the same framework would work if you consider also like signals from other sensory areas? So you have to integrate like auditory signals or visual stimuli.
Let me take one question at a time. So first question was about noise correlation, right?
Yeah.
Well, I'm not sure if I should tell you everything, because that's the homework and the thinking part. But I think it's a pretty easy exercise to figure out how noise correlation would affect the population responses. Imagine if noise correlation is 100%, that means two neurons would have exactly the same firing, right? So two neurons would give you the same information, exactly the same information. Then in this case, two neurons works just as one neuron, right? and probably you can guess whether noise correlation help you or not in this case, right?
Yeah.
Okay. Yeah. So I guess I will leave the rest to you. Maybe you can try to run some simulation to figure out how noise correlation would affect the population response. I mean, the performance of the population. All right. And what's your second question again?
Second question, please ask again, maybe.
Yeah, the second question. Can you ask again?
What do you think would happen if you consider also signals from other sensory areas? So, if, for example, you have to integrate auditory inputs and visual inputs instead of having only visual inputs as we're having now.
Right. That's a very good question, and a lot of people are studying the multimodal decision making, and in terms of theory, they shouldn't really be treated differently, like the auditory and the visual information should be just treated as two channels of probabilistic evidence. Then you should integrate the information together for decision making. But in the brain, it's more complicated. Depending on the modality, maybe one channel carries more information than the other. Maybe one channel carries information faster than the other. Then combining this information from different channels together is actually a subtle job. And many studies have shown that brain might adopt different strategies when you are dealing with different modality. For example, a study done by Yonggu, also from our institute, found that when the monkey is asked to judge self motion, both based on both the vestibular signal and the visual signal, it looks like because the vestibular signal is both faster and more accurate, the monkey relies on the vestibular signal more, and because the vestibular signal has and because the vestibular signal actually arrives to LIP a little faster than the visual signal, they found if you artificially make the visual signal appear sooner, then you could then the monkey could actually perform better in this case. So that's an example that you can manipulate the synchrony between two signals to achieve a better performance.
Okay, thanks.
I think we have a question, Veronica. Yeah.
Thanks. I wanted to ask, so you use the Drift-Diffusion model to model the activity of LIP neurons, right, but not for MT neurons. So since those also seem to accumulate evidence, I was wondering if it makes sense to use a Drift-Diffusion model also for those.
Yes. Yes, that's actually what I was trying to show you in this figure. What happened? Yeah. So in this figure, you can see the MT responses plotted below, and here the color coding is the same. So orange is for the motion coherence 51.2, and the MT responses are pretty much the same during the whole period. we don't see the same kind of accumulation process in MT comparing to LIP. So we believe MT just provides a representation for the raw sensory input, which can be used by LIP neurons. So yeah, so that's the evidence that MT itself is not doing the evidence accumulation. It contributes to it still contributes to decision making, of course, but it's not the area where the decision is made.
Thank you.
That's it. Okay.
Any other questions? Azel? Okay, it seems that we have asked all the questions.
That's wonderful. Okay, thank you. Thanks the Barso sites, and I think we have a great session. Right, we need to go for dinner now. Right.
Okay.
Okay.
Lunch for us.
You guys go for lunch. All right.
All right.
You.
See you later.
Thank you.
Thank you.
See you. Yeah.